Okay, hi all my students. Today teacher would like to continue our lesson about photosynthesis. But as you know that photosynthesis is the process by which light energy is converted to chemical energy that is used to synthesis complex organic molecules. For example, sugars from carbon dioxide in the air. Photosynthesis occurs in green plants, algae, cyanobacteria, and certain photoautotrophic bacteria. Chloroplasts are found in green plants and algae. These green plants and algae contain chlorophyll pigments, so that they will carry out photosynthesis. Then oxygen is released, used for cellular respiration in aerobic organisms. Now, before we go into pigments and photosynthesis cycle, I would like you to know about autotroph too. Okay, so autotrophs here I present about autotroph. Okay, so autotroph is an organism that uses energy from light or oxidation of inorganic substances to synthesize complex organic molecules from carbon dioxide and other inorganic substances. Autotrophs can synthesize their own food. They are producers in the biosphere. There are two types of autotroph, which are photoautotroph and chemoautotroph. So, right, in autotrophic nutrition, the organisms autotroph make use of inorganic sources of carbon, mainly carbon dioxide in the air, to synthesize complex organic molecules. So, here you got two kinds of autotroph types: photosynthesis and chemosynthesis. So photosynthesis is photoautotroph. All right. So I already mentioned this. So plants, algae, cyanobacteria, and some photoautotrophic bacteria use water as a source of hydrogen ions in photosynthesis, and oxygen is released. And then for some bacteria, for example, anaerobic green sulfur bacteria chlorobium, purple sulfur bacteria chromatium, use hydrogen sulfide as a source of hydrogen, and then sulfur is deposited. So here about chemosynthesis or chemoautotroph. If you can read there, is the process whereby the organisms chemoautotroph use carbon dioxide as an inorganic carbon source. A chemoautotroph is an organism obtain energy from oxidizing inorganic chemicals to synthesize complex organic molecules using carbon dioxide as an inorganic carbon source. Energy to drive the synthesis of organic compounds can be obtained by oxidizing inorganic chemicals such as sulfur here, iron 2, ammonium nitrite, hydrogen, and hydrogen sulfide. So chemoautotrophic play important roles in biogeochemical cycles, means they return back the minerals into the ecosystem, right? So via chemoautotrophic bacteria. So we continue the lesson. Okay. As you know that you are using for photosynthesis, okay, if we want to study the pigments inside the leaf, because leaf contain chloroplast, so you need to conduct this practical. First, separation of leaf pigments is carried out by a technique called paper chromatography. So we will conduct this later when we already in the school. Okay, we will conduct this experiment. So the leaf is first homogenized and acetone is added to dissolve the pigments. So first you need to homogenize. You need to crunch the leaf, all right, until it become a paste like that, leaf paste, and then you add acetone because acetone to dissolve the pigments. Then the mixture is filtered. The pigments are spotted onto a chromatography paper, then which is later dipped into a solvent. So here, how you dip into the solvent, you can see solvent level here, and then this is part of concentrated leaf pigments, and this is chromatography paper for sure, of course. Okay, so what is the function of solvent actually? So function of the solvent is to dissolve the pigments. All right, so each pigment has a characteristic color which forms a band on the chromatography paper. So from here, how many pigments peaks can you see? Alright. So from here, how many pigments can you see? Supposed to for pigment peaks can you can see from here. Okay. Mm-hmm. Okay, then so 1, 2, 3, 4. So this is each pigments that you can observe on the chromatogram. So it is chlorophyll B, chlorophyll A, xanthophyll, and carotenoids. Okay, then now, okay, so this should be the result you need to get in experiment 6 later. Okay. Mm-hmm. Okay, so what else? Okay, here. Now, from previous lesson, you already know about absorption spectrum. Okay, so now today, uh, so what you can tell me? Which color do all the three pigments show maximum absorption? From here, all right. So which color do all the three pigments show maximum absorption? It's supposed to be, if you can see, it is a blue color. The blue color, the three pigments show maximum absorption. Okay, okay now I continue. Hold on. Okay, so now action spectrum is what? Action spectrum is a graphical representation which indicates rate of photosynthesis at different wavelength of light. So here I already moved the slider and you can see and you now can observe the peaks of the action spectrum. What is the range of wavelength at which rate of photosynthesis is high? Okay, from here, from the graphs, you can see there is two ranges. All right, so from 480, 450 nanometer to 480 nanometer. Okay, then the second range is 680 nanometer to 700 nanometer is the range of wavelength. The rate of photosynthesis is high. Remember, absorption spectrum to show the wavelength of the colors of the greatest absorption, while the action spectrum to show rate of photosynthesis. Okay, so rate of photosynthesis is higher at this range, 450 to 480 blue, and at the red part, 680 to 700. Now, okay, what is the range of wavelength which rate of photosynthesis is low? As you can observe here, it is 490 nanometer to 550 nanometer. Then if you can compare amount of absorbance and amount of photosynthesis, so these spectrums, You need to observe the difference between absorption spectrum and action spectrum. What is the difference between these two spectra? Okay, so if you can see, absorption spectrum indicates amount of absorption at different wavelengths, while action spectrum indicates the rate of photosynthesis at different wavelengths. Right. Then what is the difference between the peaks of absorption and action spectra? It is very similar to the combined shapes of the absorption spectrum of the individual pigments. Actually, between the peaks of absorption spectra and action, actually it is combined shapes of the absorption spectrum of the individual pigments. You combine everything, B, carotenoids, A, and you will get absorption action spectrum, right? So why is the peak of action spectrum wide? Because action spectrum peak wider than absorption spectrum, because as mentioned before, it is the sum of all the pigments in absorption spectrum. Okay, so that's it. So for conclusion, today's lessons. Okay, paper chromatography technique is used to separate different pigments of a leaf so that each pigment is identified by the characteristic colored peak formed on the chromatography paper. The amount of absorption of the pigments and the rate of photosynthesis at different wavelengths of light can be graphically represented. So that absorption spectrum indicates amount of absorption at different wavelengths of light, means that while the action spectrum indicates the rate of photosynthesis at different wavelengths of light. So chlorophyll A and B absorb blue and red light, while carotenoids absorb only blue light. So from the absorption spectrum and action spectrum graph show peaks and valleys. So from if you already catch up all the points there, so we next lesson we will learn about photosynthesis, which photosynthesis is a process in which carbohydrate synthesized by using sunlight, carbon dioxide, and water. It occurs in the thylakoid membrane of the chloroplast. Remember that membrane of the chloroplast contains many pigments such as chlorophyll A, B, and carotenoids. So this is suitable for the light reaction. So these pigments are grouped together to form photosystems. So there are two types of photosystem, photosystem 1 and photosystem 2. Both these systems absorb light energy and convert it into biochemical energy. Okay, as I already covered the components of photosystem, and the details of photosystem 1 and photosystem 2 before. So by right, you can differentiate these photosystems and functions of pigment in the photosystems, absorption spectrum and action spectrum of the pigments already covered. So that's all for today. So I hope I can broadcast in YouTube later and we'll let you know. Okay, that's all. Bye.